on proto-oncogenes. Well, let's go to the content table, which includes proto-oncogenes, the transfection assay, RAS protein, how proto-oncogenes are converted to oncogenes. Now, before moving on to the topic, let's first understand what is a cancer. Well, cancer is a disease in which some of the body cells grow uncontrollably and spread to other parts of the body. These are the various types. There are various types of cancer, such as breast cancer, skin cancer, prostate cancers, and etc. Well, cancer is caused by the change to DNA, as these changes occurs in the section of DNA which is known as genes. Or in other words, we can say that the cancer occurs when proto-oncogenes is mutated. It forms oncogenes, which are basically responsible to cause cancer. So now let's move on to the first one, which is proto-oncogenes. Well, proto-oncogenes are the genes that are involved in normal cell growth, cell divisions, and which help cell to stay alive. They are the healthy genes which are found in cells, which contains the necessary information of your body to make the necessary information of your body to make the protein responsible for cell growth and cell division. But when proto-oncogenes are mutated, they make too many copies of itself and turn on when it's not supposed to be. At this point, they become oncogenes, which causes cancers. Oncogenes are the key genes to the key genes of causing cancer. The V-onc, or we can say viral oncogenes, and C-onc, or cellular oncogenes, are the mutated form of proto-oncogenes. To study the gene functions and regulations of proto-oncogene, transfection assay is one of the most powerful analytical tools used. So now let's look at what is transfection assay. Well, transfection assay is an assay used to identify DNA sequence which is capable of basically transferring or transforming normal cells to the cancerous cells, or we can say it is used to identify mutant proto-oncogenes in the given tumor. As to perform this assay, the DNA is extracted from the cancerous tissue. Uh, then, uh, then they are fragmented into small pieces, and these pieces are then joined to the bacterial DNA, which acts as a molecular markers. Then these marked DNAs are transfected, or we can say introduced. Uh, they're introduced in the actively growing cell cultures. Later, to recognize these, whether there are markers or not, small lumps or foci are formed on the soft agar plate. Uh, Later, the DNA is extracted from those cells and is screened to check for the molecular markers that are linked to the original fragments. Once it's identified, the DNA is then rested, or we can say confirmed, for its ability to induce cancerous state. So this is known as transfection assay. Now, next one is the RAS protein. Well, RAS protein is a proto-oncogene that are frequently mutated in human cancer. In mammalian cells, there are three main RAS protein which closely express. That is HRAS, which is known as Harvey strain, which encodes codon 12. KRAS, which is also known as Christian stain, and NRAS, which is also known as neuroblastoma stain, as these strain promotes oncogenesis. The Weinberg's research teams found that the CHRAS oncogenes from a cancerous bladder cells, as the DNA was extracted from those cells and transfection assay was performed. Later, the mutant ChRNA gene found to be does not synthesize abnormally large amount of protein due to the substitution mutation of codon 12, which deteriorates the ability of the mutant ChRAS gene to hydrolyze its substrate. The deterioration which causes the mutant protein an active signaling mode, which transmits signal and stimulates the cell division in an uncontrolled way. So now let's look at the regulation. So the first is the regulation of normal RAS protein. So what happens in normal RAS protein is that so over here it's an extracellular signal is transmitted to the plasma membrane, which influence the state of RAS protein. The inactive RAS protein in the cytoplasm gets activated by the phosphorylation of the bound GDP. The GDP then gets converted to GTP and activates the RAS protein, which induces a signal to the nucleus, to the nuclear membrane. Later, this signal regulates the transcription of the genes which are involved in a cell division, and the cell division occurs in a controlled fashion. Once the signal is transduced, the active RAS protein becomes inactive by dephosphorylation of GTP, and again it gets activated when extracellular signal is transduced. So this is what happens in normal RAS. Whereas in mutant RAS protein, an extracellular signal has no influence on the state of the mutant RAS, as the mutant RAS protein remains in an active state only when transfused and when an sorry it's in a mutant state only when it's an active state only when transduce an established signal to the nucleus, as these signals then cause the genes involved in a cell division to be transcribed inappropriate, and then cell division occurs in a uncontrolled fashion. So this is the mutant RAS. Now let's look at how proto-oncogenes transfer to the oncogenes. But before moving on, now let's look at the protein products of the proto-oncogenes. Well, the proto-oncogenes are the normal cellular genes that regulate cell growth and differentiation. They often encode proteins such as growth factors, protein kinases, membrane-bound G proteins, and etc. Well, the examples of growth factors are V-onc and C proto-oncogenes, which alters the growth factor level. Then the protein kinase is an enzyme which adds it's a protein which adds the phosphate group to the target protein and modifies the functions. And the example for membrane associated G protein is a well-known RAS protein. Okay, so now let's look at what are the factors which influence proto-oncogenes to form oncogenes. In this, the first is the point mutation. It is a mutation occur in a genome when a single base pair is added, deleted, or changed. As this mutation increases the activity and expressions of the gene, which in turn increases the amount of gene product. As the example for point mutation is RAS protein, as we have previously looked in the mutant RAS. The second is the deletion, where due to deletion, the activity of the gene is altered. The third is the gene amplification. Well, in this, some tumors have multiple copies of proto-oncogenes. So even if one proto-oncogene is expressed, it stimulates other copies as well. So there is a well-known example for this, and it is uh, multiple copies of RAS proteins are found in the mouse it's adrenocortical tumor. Next, the fourth one is the chromosomal rearrangement. In chromosomal rearrangement, CML, which is also known as chronic melanogenous leukemia, which is also known as Philadelphia chromosome, is a well-known example as it is associated with the aberration of chromosome 22. Sorry. So these are some factors or changes we can say which influence the proto-oncogenes to convert to oncogenes and cause cancer. These are the references, and thank you. Sorry. In RAS, uh, it it would be, so 1 second. Uh, I think HRAS HRAS maybe would be mutated. Yes, sir. Which enzyme? So I have no idea. Proto-oncogenes. I think it's dominant, sir. Yeah, dominant.
Yes, sir. Sir, it's visible. Hello, sir. Sir, it's visible. Hello. Good evening, everyone. Myself Sanjeevani Pad. हाँ sir. Good evening, everyone. Myself Sanjeevani Padwar, and my today's presentation topic is cancer cells and their properties. So let's see first content. Uh, so what is cancer? Cancer is a disease in which some of the body cells divide rapidly, abnormally, and uncontrollably, and they can invade nearby tissue and spread to other parts of the body. Cell which is undergoes rapid, abnormal, and uncontrolled growth at the cost of remaining cells are called a neoplastic cell. The growth of resulting form of the division of such cells are called neoplastic growth or tumors. In another word, tumor is an abnormal mass of tissue resulting in uncontrolled division, that is cancer. Tumor cells may be carried by the bloodstream or the lymphatic system. They can also carried by direct penetration to other parts of the body, where they may include secondary tumors. Most cancers form tumors, but not all tumors are cancerous. Tumors are classified into two types. First is benign tumors, and second is a malignant tumor. So let's see one by one. So first, benign tumors. Benign tumors are not cancerous. They do not spread to other part of the body. These tumors are localized at the particular location in the body and small size of nuclei. Benign tumors usually contain well differentiated cells. Then second one is a malignant tumor. Malignant tumors are cancerous, means that they will grow quickly and spread to other normal tissues of the body. Malignant tumors spread into and or invade nearby tissue and can travel to distant place in the body to form new tumors, and this process is called as a metastasis. Malignant tumors usually contain undifferentiated cells, often with large nuclei. Then types of cancers. Cancers are classified into four groups or types, which is first one is a carcinoma, second is a sarcoma, uh, third one is a lymphoma, and fourth one is a leukemia. So let's see sarcomia. What is sarcomia? Sarcomia, carcinomas are tumors made up of principally of epithelial cells of ectodermal or endodermal from solid tumors. They are the most common type of cancers. Examples of carcinomas include breast cancer, lung cancers, and etc. Sarcomas. Sarcomas are tumors made up of principally by connective tissue cells, which are mesodermal origin. They are solid tumors growing from connective tissue, cartilage, bone, and muscles. Third one is a lymphomas. Lymphomas is a cancer that beginning in the lymphatic system. Lymphomas are cancerous in which there is an excessive production of lymphocytes by the spleen. The lymphatic system is a network of vessels and glands that helps to fight the infection. Hodgkin's disease is example of lymphoma. Then fourth type is leukemia. Leukemias are neoplastic growth of leukocytes and are characterized by the. Then next one is the property analysis. So let's see one by one. Property is immortalization. Immortalization on external factors in contrast of growth factors. Reasons by small scale. Examples human and circumstances is called like inhibition. Normal cells in a culture stop growing when their plasma membranes come into contact with one another. When two normal cells come into contact, one or both will stop moving and then beginning to move in another direction. This inhibition of growth after contact is called as a contact inhibition. Normal cell growth in a culture until they cover the surface as a monolayer. Cancerous cells grow in multi-layer clumps and they pile up one above the other. Transformed cells do not stop dividing after forming a monolayer. Third is the invasiveness. Invasiveness is one of the most important characteristics of a transformed cell. Cancerous cells are invasive. The invasiveness could be the result of change in plasma membrane or protease released by the cells. Fourth one is the loss of anchorage dependence. Normal cells show the property anchorage dependent growth, and most normal cells must be attached to the rigid substratum in order to grow. Therefore, normal cells are not able to grow properly in liquid and semi-solid media. Tumor cells exhibit anchorage dependent independent growth. Tumors do not require rigid substratum for growth. They are able to grow in liquid or semi-solid media. Then transformed cells can grow even when they are not the. Then next property is defective electric communication. Electric connection normally occurs between individual cells. In some cancer cells, however, it has been reported that such connections are defective. The next property is lower serum requirements. Growth of normal cells in a tissue culture medium requires a high concentration of serum. It provides complex factors such as insulin, other hormones, and growth factors. Some serum growth factors resemble insulin in interacting with external receptors on the cell membrane to regulate biochemical activities within the cell. Then seventh property is increased sugar transport. Tumor cells consume much more glucose than normal cells because they have to grow and multiply. There is a great increase in the rate of sugar transport across the surface cell membrane after transformation. Eighth property is increasing in negative surface charge of cell membrane. That is compression of surface membrane charge by microelectrophoresis have been made between normal and malignant cells. Then in malignant cell, amidoid mobility is usually higher, indicating increasing in a negative surface charge. Ninth properties is increased secretion of proteolytic enzyme. Large amount of proteolytic enzymes are secreted by all types of cancers, expected those of blood-forming tissue. The cancer cell secreted a protease called the cell factor. The cell factor acts on an inert serum protein plasmogene to form a plasmid, and plasmid remove many proteins projecting from the cell surface by enzymatic digestion and signal to cell into division. If normal cells are treated with protease, they show many of the characteristics of the transformed cells. Then, 10th one is aldolase. In most mammalian tissue, the enzyme aldolase exists in the form of the three isozymes: isozyme A, isozyme B, and isozyme C. Isozyme A and isozyme C predominate in embryonic tissue, while in adult differentiative tissue, the B isozyme is a predominant. In some tumors, especially in poorly differentiated and rapidly growing cancer like certain hepatomas, isozyme B is released by isozyme A, the embryonic form. There is a small difference between normal cells and cancerous cells. These are the references. Thank you. Yes, sir. It is formation of new blood cells. Uh, Sorry, sir. Hello, sir. It is formation of new blood cells. It is important not only supporting tumor growth but uh, metastatic. Primary cell line means
Yes, sir. Sorry, sir. Sir, is my screen visible? Okay. Good evening, everyone. Myself, Aliza Parkar, and my topic for today's presentation is cancer and cell cycle. Uh, these are the table of contents. We're gonna talk about cancer, benign and malignant tumor, key differences between benign and malignant tumor, types of cancer, causes of cancer, and cell cycle. Cancer. Cancer is a disease characterized by the uncontrolled growth and spread of abnormal cells. These abnormal cells can form tumors, invade nearby tissue and organs, and spread to other parts of the body through the bloodstream and lymphatic system. Normally, the cells in the body divide and grow in a controlled manner, but cancer cells have lost the ability to control their growth and division. Benign and malignant tumor. Benign and malignant tumor are two types of growth and masses that can develop in the body. Benign tumors are non-cancerous growth that typically not spread to the other parts of the body, whereas malignant tumors are cancerous growths that can invade nearby tissues and spread to other parts of the body. These are some key differences between benign and malignant tumor. First is growth rate. Benign tumor tend to grow slowly, while malignant tumor can grow rapidly and spread to the other parts of the body. Ability to spread. Benign tumors are usually confined to one location and they do not spread to other parts of the body. Malignant tumor can invade nearby tissues and spread through the bloodstream or lymphatic system to other parts of the body. Invasion that benign tumors are contained within a capsule and they do not invade nearby tissues, whereas malignant tumor can invade and destroy nearby tissues. Cell characteristics, benign tumor are made up of cells that are similar to normal cells, while malignant tumor may have abnormal cell characteristics like irregular shapes and sizes. Then recurrence, benign tumor typically do not recur once removed, but malignant tumors may recur even after the treatment. Treatment, benign tumor are usually removed surgically, while malignant tumor, it involves surgery, chemotherapy, radiation therapy, targeted therapy, or combination of all these. The types of cancer. Cancer is not a single disease. It is a complex of many diseases. And like there are 200 distinct type of cancers recognized, which can be grouped into four main types. That is carcinoma first. It is a type of tumor made up of epithelial cells of ectodermal or endodermal origin. Second is sarcomas. These are the tumors made up of complex tissue cells, which are of mesodermal origin. Lymphomas are cancers in which there is excessive production of lymphocytes by lymph nodes. And leukemias, they are neoplastic growth of leukocytes. Causes of cancer, there are several factors that can increase the risk of causing cancer in humans. Some of those causes are genetics, like a certain genetic mutation can increase a person's risk of developing certain type of cancer. Environmental factors like exposure to certain chemicals, radiations, or viruses can increase the risk of cancer. And lifestyle factors, smoking or excessive alcohol consumption, poor diet, and lack of physical activity can also cause cancer. Cell cycle. The cell cycle is a process by which a cell grows and replicates its genetic material and divides into two daughter cells. In every cell cycle, all chromosomes must be duplicated and a copy of each should be distributed to both the progeny cells. The cell cycle is divided into four phases: G1 phase, S phase, G2 phase, and M phase. Then it consists of checkpoints. Like checkpoints at different points in the cell cycle are the control points at which the cell cycle is arrested, like if there is damage to the genome or the cell cycle machinery. In this, it allows the damage to be repaired, or if it is not, the cell is completely destroyed. These processes are necessary because, like it prevents the possibilities of damaged cells dividing in an unprogrammed way, that is, from becoming cancerous. Then, as cell proceeds through the G1 phase, it prepares for DNA replication and chromosome duplication in S phase. Then the major checkpoint, G1, it determines whether the cell is able to move or it should continue in the S phase. Like the cell stays in the G1 unless it grows big enough and the environment is favorable. Another major checkpoint is G2 to M checkpoint. It occurs at the junction between the G2 and M. Unless all the DNA have been replicated, the cell has become big enough and the environment is favorable, the cell cannot enter the mitotic phase of the cell cycle. Then the third checkpoint, it occurs during the M phase. In this, the chromosome must be attached properly to the mitotic spindle to trigger the separation of chromatids and to complete the mitosis. Then the protein, which is cyclin, and enzyme, cyclin-dependent kinase, these are the key components in the regulatory events occurring at the checkpoints. At G1 to S checkpoint, two different G1 cyclin CDK complexes form, which results in activation of kinase. Kinase catalyzes the series of phosphorylation, that is, addition of phosphate group of cell cycle protein. It affects the function of protein and it leads to transition into the S phase. This similar process occurs at G2 to M checkpoint. G2 cycling binds to CDK to form a complex. Then, until the cell is ready to enter mitosis, phosphorylation of the CDK by another kinase keeps CDK inactive. These are my references. Thank you. Yeah, you are. Cycling, I don't know. M phase. Division in M phase. Uh, मतलब increase in nucleus size. G1 phase. Yes. G1. G. Interphase. Interphase. हाँ. Uh, the cycle will keep. So what did you ask? सर हो गए 4 से ज्यादा क्वेश्चन जाने दो. G1 to won't proceed. If like the cell isn't grown large enough and if the environment is not favorable, it won't proceed further. Nice. 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 करता है. सर मेरा आंसर भी करेक्ट है ना. हाँ, क्या आधा ही मार्क्स देना. नहीं sir, नहीं. Sir, shall I stop sharing? Thank you, sir.
सर हुआ है मेरा.
Yes, sir. Sir, my screen is visible. Wait, sir. Sir, is visible? Yes. So, good evening, all. My name is Neha Zungare. So, today's my topic is which I'm going to present you all is Knudsen two-hit hypothesis. So, here is the content of my slide. So, he is the scientist Alfred Knudsen who proposed the two-hit mutation of retinoblastoma gene in 1971. He proposed that the retinoblastoma is a cancer caused by two mutational event occurs in alleles. So, what is the retinoblastoma? The retinoblastoma is a cancer that starts in the retina, the very back part of the eye. It's a childhood cancer of the eye which developed during the period from the birth to age 4 years. This tumor suppressor, retinoblastoma gene, is the first discovered. The location of this gene is a nucleus on chromosome 13. So, uh, here we can easily differentiate between the normal eye and the mutated eye. The healthy eye is we can see no tumor is we can see in a retinoblastoma mutated eye. We can easily see the tumor is formed. So, there are retinoblastoma can be subdivided into two types, mainly the sporadic retinoblastoma and the hereditary retinoblastoma. So, in sporadic retinoblastoma, an eye tumor develops spontaneously in a patient from the family with no history of the disease. In this case, a unilateral tumor develops in one eye only. This mutation occurs in a somatic cell. The child is born with the two wild types copies of the retinoblastoma gene, which genotype is a homozygote. The first mutation producing an heterozygous retinoblastoma cell, and the second mutation in the cell giving rise to the uh, uh, homozygous genotype that result in a eye tumor development. In hereditary retinoblastoma, the susceptibility to develop the eye tumor is inherited. In this case, bilateral tumor develops in a typically multiple eye tumor involving both eyes. This mutation occur in germinal cell. In hereditary retinoblastoma, the patient inherit one copy mutated gene that is the uh, heterozygous, which is the heterozygous, only a single additional mutation of the retinoblastoma gene is an eye cell is needed to produce an homozygote type allele that would result in tumor formation. So here we can see in a sporadic retinoblastoma, the child is born with the two wild type copies of the retinoblastoma gene, which is genotype is a homozygous, where the cell growth is a normal. The first mutation producing an
heterozygous cell. Here, the one retinoblastoma gene is mutated. The cell growth is also normal in a first hit of the mutation, but the second hit mutation cell is giving rise to the uh, homozygous genotype, which is result in loss of growth control and leads to tumor of eye. This type of cancer is a very rare. In hereditary retinoblastoma, we can see the child is born with a one mutated retinoblastoma gene. The cell growth is normal. Here, the single additional mutation of the retinoblastoma gene is need to produce the homozygous genotype that result in tumor formation and loss of growth control of the cell. So the function of the retinoblastoma tumor suppressor genes, the PRB is involved in regulating the cell cycle at the G1 to S checkpoint. G1 to S checkpoint, the two CDK cycline complexes form during the G1 phase, and CDK4 and CDK4 cycline D and CDK2 cycline E is formed. These complexes cause the progression to the S phase by catalyzing a series of the phosphorylation of all cycle, which controlling the proteins including PRB. PRB is found in a complex with the transcription factor E2F, and when the PRB is unphosphorylated, then the activity of E2F is inhibited. So, then when PRB becomes phosphorylated, the inhibition of E2F activity is removed, and then the now active transcription factor turns on the transcription of gene of DNA synthesis. As the cell begins a DNA synthesis, the cyclin D and cyclin E are degraded, and the cyclin A is made, and the CDK2 and cyclin A complexes form and activates the DNA replication. And after the S phase, the retinoblastoma protein is dephosphorylated and again entering the E2F inactive. So the conclusion of the two-hit mutation hypothesis, which is according to Knudsen model, the retinoblastoma mutation is the recessive because cancer develops only if both alleles are mutated. If one mutation is inherited through the germline or tumor formation, requires only a mutational event. The remaining wild type alleles in any one of the cell is that particular tissue. A person to cancer but does not cause the directly. A second mutation is required for the loss of heterozygosity. So these are my references, and thank you. Hello, sir. Sir. Okay, sir. Yes, sir. That's a two-hit mutation. हाँ सर, डोमिनेंट होता तो वो कॉज करता ना कैंसर, करता मतलब कॉज करता वो। यस डोमिनेंट होता तो 1 हिट लगता. नहीं सॉरी 2 हिट लगता सर. 2 हिट है सर. व्हेन रेसेसिव है सर. स्पोरेडिक रेटिनोब्लास्टोमा में फर्स्ट होमोजाइगोट अलील्स रहता है. सो उसको म्यूटेड होने के लिए 2 हिट लगता है. बट हेरिडिटी में वो फैमिलियल होता है और पहले से म्यूटेटेड आता है. सो उसको एक ही हिट की जरूरत होती है और ट्यूमर को कॉज करने के लिए. थैंक यू सर.
Sir, it's my screen visible? Yes. Good evening, one and all here. Myself, Nidhi Mahale, and today's my presentation topic is tumor suppressor genes. So let's start with the. These are my contents throughout which I am going to cover throughout the presentation. Let's start with some basic concept. What is cancer? Cancer is a group of disease caused by uncontrolled multiplication of abnormal cells in the body, a process called as neoplasia. It is usually caused by mutations in somatic cell genes that regulate cell growth. Almost every tissue in the body can produce cancer. Some even generate many different types of cancer. However, cancer mostly occurs in cells that divide and reproduce more than other cells. Causes of cancer: it is caused by benzene or other chemicals, due to drinking excess alcohol, excessive sunlight exposure, some kind of genetic problems, obesity, radiation, and viruses. Uh, uh, in order to support the growth of an organism, cell needs to grow and divide. But cell needs to grow and divide. But after some point of time, cells also leads to a death. That is means cells leads to a cell death. So there are two categories of gene. One which promotes cell division, uh, proliferation, and growth, called as proto-oncogenes, while other category is tumor suppressor gene, which prevent the excessive growth of cell or tissues. So any uncontrolled mutation or uncontrolled proliferation would be a cause of a cancer. Now, if any gain of function of mutation in proto-oncogene or um, loss of function of mutation in tumor suppressor gene leads to uncontrolled division, which would ultimately result to or leads to an cancer. Now, it is important to understand any loss of function mutation in tumor suppressor gene would lead to a cancer. So tumor suppressor name itself suggests the formation of tumor. Tumor suppressor gene or anti-oncogene is a gene that regulates a cell during cell division and replication. Products of tumor suppressor gene are tumor suppressor proteins. They regulate cell growth by breaks to cell proliferation. That means they are growth inhibitory proteins. Failure of growth inhibition is seen in carcinogenesis. Reason for that is loss of gene function of these genes, which leads to a carcinogenesis. These are some most common tumor suppressor genes. RB retinoblastoma gene, which is located in nucleus on chromosome 13, which causes human tumors. It is called retinoblastoma and osteosarcoma. T53, T50, T53, which is located in nucleus on chromosome 13, on chromosome 17, which causes human tumors like lung cancer, colon cancer, neck and breast cancer. APC, which is located in cytosol, which causes colon cancer, and some other types of genes, that is WT1 and 2, and BRCA 1 and 2, which both are located in nucleus. WT gene causes Wilson tumor, and BRCA gene causes breast cancer or ovary cancer. Now, before we understand the concept of tumor suppressor gene or RB gene, let us see what does loss of heterozygosity means. In this tumor suppressor gene, when both alleles of genes are normal, there is normal function of this particular gene or tumor suppressor gene. But if one of the allele is abnormal or inactive, this particular state is called as heterozygous state, still the function is maintained, so that it is normal function of tumor suppressor gene, even though it is heterozygous. All we need is only one normal allele of a gene. Now both of these alleles are abnormal and both of these alleles are inactive. Then there is loss of function of tumor suppressor gene that causes proliferation of cells, resulting in formation of tumor. This condition of loss of heterozygosity is mainly caused due to either deletion or mutation in tumor suppressor gene. Tumor suppressor genes are categorized into two forms: gatekeeper and caretaker. Gatekeeper is which directly suppress the growth. Caretaker, which maintains overall genetic stability. Gatekeeper contains cell cycle regulatory genes, checkpoint control genes, and apoptosis related genes, whereas caretaker contains DNA repair proteins. Types of tumor suppressor genes, one of which is retinoblastoma. Retinoblastoma, it is first discovered gene, tumor suppressor gene. It is located on chromosome 13. Retinoblastoma is a childhood tumor with inactivation of this gene. It plays a key role in regulation of cell cycle, and therefore it is also called as governor of the cell cycle. Knudsen two-hit hypothesis. Two mutations involving both the alleles of tumor suppressor gene is a basic requisite for development of tumor. Retinoblastoma can occur as hereditary or sporadic form. We will see in short how Knudsen two-hit mutational event takes place. Here, in case of hereditary retinoblastoma, what happens at the birth? There is already one mutation. So the first mutation is at the time of birth. So the first hit is inherited mutation, whereas further second hit occurs any time after the birth. Once you have second hit, that is both alleles are abnormal or inactive, results in formation of tumor. Whereas in sporadic retinoblastoma, at the time of birth, both alleles are normal. So first hit occurs as acquired mutation, And second hit also is acquired mutation. So once both alleles are abnormal, results in uh, formation of tumor. P53 gene. It is normal functioning gene where which prevent propagation of genetically damaged cells. When DNA is damaged, P53 upregulation transcription of genes that arrest cell cycle and repair DNA. Cell cycle arrest is immediate by P53 dependent transcription by CDK inhibitor P21. If DNA cannot be repaired, P53 induces apoptosis. These are some references. Thank you. Sir, BRACA 1 and 2. Sir, I didn't understand your question. Yes. Wait. Yes. No idea, sir.
Yes, sir. Is it okay now, sir? Okay. Good evening, everyone. Good evening, sir. Myself, Vidhi Shirsagar, and my topic of presentation is proto-oncogenes. This is the content of my presentation. Before move, it is not visible. Is it now? Is it visible? Good evening, everyone. Good evening, sir. Myself, Vidhi Shirsagar, and my topic of presentation is proto-oncogenes. This is the content of my presentation. Before moving to proto-oncogenes, we will first discuss what are genes and how these genes are related to cancers. There are four main genes when altered lead to cancer formation. First is proto-oncogenes. Proto-oncogenes normally stimulate cell growth and proliferation. These proto-oncogenes, when undergo mutation, lead to formation of oncogenes. Oncogenes are present in cancerous cells and they might be active at inappropriate times, which lead to cancer development. Second is tumor suppressor genes. Normally, tumor suppressor genes inhibit cell proliferation. When these tumor suppressor genes undergo mutations, the cell in which these tumor suppressor genes are mutated lost their inhibitory functions. Third is the microRNA genes. MicroRNA genes normally targets the mRNA of other genes and inhibits their translation. Fourth is the mutator genes. Mutator genes normally ensures the fidelity and replication and maintenance of genome integrity. Now, what are proto-oncogenes? Proto-oncogenes are the healthy genes found in the cells. A gene, proto-oncogenes are normally involved in normal cell growth. Mutation in proto-oncogenes may cause it to become an oncogenic, which can cause the growth of cancer cells. There are many proto-oncogenes. Each one is responsible for making a protein involved in cell growth, division, and other processes. Oncogenes. Now, what are these oncogenes? As we all know, the oncogenes are the mutated form of proto-oncogenes, which are normally present in cancerous cells. Oncogenes is a gene whose action stimulates unregulated cell proliferation. Tumor viruses, these oncogenes are present in tumor viruses, which cause neoplastic cells. Now, what are these tumor viruses? Tumor viruses are the viruses which contains oncogene in their genomic material. When this tumor viruses infects a cell, they transform the normal cell into a cancerous state or a neoplastic state. This tumor viruses might be of DNA origin or of RNA origins. The tumor viruses which contains RNA as their genomes are called as RNA tumor viruses, and the tumor viruses which contain DNA as their genome, they are called as DNA tumor viruses. Viral oncogenes, retroviruses and oncogenes. Retroviruses mostly contains RNA as their genomic material. RNA tumor viruses are all retroviruses. Oncogene carried by RNA tumor viruses are altered form of normal host cell gene. To understand this, we will first look at the life cycle of retroviruses. As the retrovirus infects the cell, the envelope which is covering the caspid layer left behind during the infection, and the caspids enters the cell. Then the uncoating occurs. After uncoating, the viral RNA is released. This viral RNA is converted to proviral DNA with the help of enzyme reverse transcriptase. This proviral DNA is then integrated into the host cellular genome with the help of enzyme integrase, and the integrated form of proviral DNA and host chromosome and is then transcribed to form an mRNA, and which is then translated to form a new progene virus to form proteins for new progene viruses. Now, this new form progene viruses contains the integrated form of DNA. Structure of retroviruses. Retrovirus structures, as we all know, the virus is made up of protein core, which we call as caspids. There are two, this virus structure present over here is of RSV, Rous sarcoma virus. Virus structure is made up of protein core, which we called as caspids. There are two copies of plus sense RNA genome. Now, what is plus sense RNA genome? Plus sense RNA genome is the genome which can be directly, plus sense RNA is the mRNA which can be directly translated into protein. If a virus contains a negative sense mRNA, then this negative sense needs to be converted into positive sense RNA, and then this RNA is translated to protein. The virus contains an envelope which is made up of glycoproteins. There are three protein coding genes. First one is GAG gene. GAG gene codes for protein particles of viruses. Pol gene codes for a Pol gene codes for an enzyme called as reverse transcriptase, and ENV genes codes for a envelope envelope glycoproteins. Viral oncogenes. An oncogenic retrovirus carries viral oncogene in its genome. Viral oncogenes are responsible for many different cancers. These viral oncogenes are named as the which cancer they caused with the addition of prefix. For example, Rose sarcoma virus leads to carcinoma. So here it is named as viral SRC. Transducing retroviruses, bacteriophages that have picked up the cellular genes are said to transduce the genes to other cells. Now, what are these bacteriophages? Bacteriophages are the viruses that infects bacteria, and transducing is the process of integration of viral DNA into the cellular DNA. Non-transducing retroviruses do not carry oncogene. If the cellular oncogene was picked up was of oncogenic, the modified retrovirus is oncogenic. If the cellular gene picked up was proto-oncogene, modified retrovirus may still be oncogenic if increased expression of proto-oncogene can cause oncogenesis. Now, what are these cellular proto-oncogenes? Cellular proto-oncogenes were discovered in the middle of 1970. The discovery led to the conclusion that these genes are present in normal animal cells DNA sequence. These are closely related to the viral oncogenes. Now, what is oncogenesis? Oncogenesis is a dominant mutation. Now, what is oncogenesis? Oncogenesis is the process where the normal proto-oncogene is converted to oncogene. It is a dominant mutation. Now, what is meant by dominant mutation? Dominant mutation means only a single mutation event is necessary in a homologous pair to convert it from proto-oncogene to oncogene. If oncogenes are carried by viruses, these oncogenes are called as viral oncogenes. Cellular oncogenes. Now, what are cellular oncogenes? Cellular oncogenes resides in host chromosomes oncogenes. Transducing retroviruses carries altered form of cellular oncogenes. Now, there is a difference between proto-oncogenes and viral oncogenes. Proto-oncogenes contains introns, whereas viral oncogenes does not contain introns. These introns are spliced during the transcription process. Now, what is transformation? Transformation is the process where the normal cell is transformed to a cancerous state. And what is transfection assay? What is transfection assay? Transfection test is done to identify DNA sequence capable of transforming normal cell into a cancerous cells. Okay, to understand this transfection process, we will first look at the diagram. This transfection process in which the tumor cells containing DNA were marked with markers. This marked tumor DNA were then isolated into normal cell, transferred into normal cells, and were allowed to grow for descendant conditions, allowed to grow for next generations. Oncogenes were integrated into the cell and transformed the descendant into cancerous cells. Then the descendant cells were isolated and the colonies were observed. By the observation of the DNA of the descendant cells, it was observed that oncogene were transformed from the primary cell to its progenies. Protein product of proto-oncogenes. Proto-oncogene product is involved in positive control of cell growth and division. Product stimulates growth or are in pathway involved in growth stimulation. Three examples of the protein product of proto-oncogenes. Growth factors. Growth factors example is viral oncogenes and cellular oncogenes that alter the growth factor level. Second is protein kinases. Protein kinases add phosphate group to target protein. Those modify function. Example of protein kinases is SRC, membrane associated G protein. The example of it is RAS protein encoded by RAS gene. Now in this slide, we are going to discuss normal RAS protein and its regulation. Now, for the regulation of RAS protein, there is a need of extracellular signal. Normally, the RAS protein is in the inactive state. Inactive state means when it is bound to GDP. Normally, it is bound to GDP. For converting it to an active state, there should be an enzyme called as GTPase, which hydrolyzes this GTP to GDP, and these phosphate released is then added to GDP, which lead to activation of RAS protein. This activated RAS protein then sends the signal, which leads to transcription of genes involved in cell division, and cell division occurs in a controlled fashion. Now, what happens in the mutated protein, RAS proteins? As I previously said, the RAS proteins are present in an inactivated state when the GDP is bound to it. But when the RAS proteins are mutated, the GTP is always bound to it, even if they have extracellular signals or not. The GTP is always bound to it. So it leads to a continual signals which lead to transcription of proteins required for cell division, continuously transcription of proteins required for cell division, and because of an uncontrolled cellular division occurs. Now in this slide, we are going to discuss changing cellular proto-oncogene to oncogenes. There are three mechanisms involved for conversion of proto-oncogenes into oncogenes. First is point mutation. Coding region or in controlling sequence, point mutations when occurs in coding regions or in controlling sequences. Example in promoter, regulator, enhancers. The example of the point mutation, most common example is the RAS mutation. Second, cellular changes is a deletion. Coding region or parts of controlling sequence of proto-oncogene. When the deletion occurs in the coding region or parts of the sequence which control the proto-oncogenes, the example of deletion event is MYC. Third event is gene amplification. Gene amplification is the process in which there is increased number of copies of genes, result in increased amount of gene product. Example of it is RAS. These are my references. Thank you. GTPase, GTPase and GTPase or kinase, GTPase, I guess. So because GTPase is the enzymes which convert GTP to GDP, and the released phosphates then goes and binds to the inactive RAS protein which has GDP, and then there is a conversion of GDP to GTP. Yes, what's up? Okay. Yes, if the proto-oncogenes get mutated into oncogenes, it will definitely lead to formation of cancers because proto-oncogene will stimulate unregulated growth of the cell as as it will as even if there is no extracellular signal, it will send signal to the nucleus uh, to transcribe proteins which are necessary for growth of cells, because of which what happens, the cell will continuously grow and lead to formation of tumor. Okay.
Are visible. Hello. Sir, is my okay. So, good evening. My presentation topic is chromosome rearrangement and cancer. So, let's see what is chromosome rearrangement. So, chromosome rearrangement refers to change in structural organization of chromosomes in the nucleus of the cell. This change can involve alteration in number, size, shape, or position of chromosomes. Like chromosome rearrangement can occur spontaneously or may be caused by exposure to radiation, chemicals, or other environmental factors, which can lead to oncogenesis, inactivation of suppressor genes, and disruption of normal cell growth and division, contributing to development and progression of cancer cells. So here are some type of chromosome rearrangement. First type we can see is translocation. So basically in translocation, what happens is the exchange of segment between non-homologous chromosome. We will see in detail about it. In inversion, there is reversal of orientation of a segment of a chromosome. And in deletion, from the name itself, we can see that there is a loss of segment of chromosome. In duplication, we can see that the presence of an extra segment of a chromosome. So in further slide, we will see in detail about types of chromosome rearrangement which lead to cancer. So this is chromosome translocation. In chromosome translocation occurs when two different chromosome exchange parts leading to fusion of two previously separate segments. We can see here is a chromosome A and here is a chromosome B. It is divided into two parts and it is interchanged. They get fused and they create a balanced translocation. This can result in formation of an abnormal gene product that contribute to cancer development. So next is inversion. So what is inversion? A chromosome inversion occurs when a portion of chromosome is reversed. It means there is a cut between the chromosome and it is reversed and it is placed in same chromosome. This can result in alteration of gene regulation and contribute to cancer development. So the third one is chromosome deletion. So in deletion occurs when a portion of a chromosome is lost. It means one part of chromosome is removed. This can result in loss of important tumor suppressor genes leading to formation of cancer. It means if any important gene can be removed from the chromosome and it can lead to cancer. So the next one is duplication. Chromosome duplication occurs when a portion of a chromosome is copied. It means same copy of the chromosome is placed in same chromosome. This can result in overproduction of oncogenes leading to formation of cancer. So this is the last one. This is aneuploidy. Aneuploidy refers to a change in the number of chromosomes in a cell. Cells with an abnormal number of chromosomes are often associated with cancer. So, chromosomal rearrangement have significant consequences on individual health. As we can see, it is a genetic change that occurs when chromosome break and then reattach in a different way. In cancer, this type of genetic alteration can lead to formation of abnormal chromosome or fusion genes, which contribute to development and progression of cancer. Chromosome rearrangement often result in loss or gain of genetic material, which can affect the normal functioning of genes and contribute to the development, loss, gain of genetic material, which can affect the normal functioning of genes and contribute to development of cancer by which promoting uncontrolled cell growth. So these are the references. Thank you. Thank you, sir. Uh, Sir, if it become oncogenic, it will lead to cancer, right? Proto-oncogenes are make protein for body function, right? If they get mutated, it become oncogene, which can lead to cancer, sir. What, sir? Sir, it will not lead to cancer because it will suppress the formation of tumors, as the name itself says, tumor suppressor gene. Welcome, sir.
Yes, sir. Yes, sir. One second. Sir, it is visible. Good evening, all of you. And myself, Vishal Subhash Patil, and my topic name is tumor suppressor gene. This is the contents we are see. First, we will see the introduction. A tumor suppressor gene or anti-oncogene is a gene that regulates a cell during cell division and replication. The discovery of oncogenes and their ability to deregulate cellular processes related to cell proliferation and development appearance uh, first in the literature as opposed to the idea of tumor suppressor genes. When a tumor suppressor gene is mutated, it results in a loss of reduction in its function. In combination with other genetic mutations, this could allow the cell to grow normally. Then second is tumor. A tumor, also known as a neoplasm, is an abnormal mass of tissue which may be solid or fluid slip. A tumor does not mean cancer. Tumor can be benign, not cancerous, pre-malignant or malignant. Then, what is tumor suppressor gene? The tumor suppressor genes are that regulate the growth of cells. When these genes are functioning properly, they can prevent and inhibit the growth of tumors. When tumor suppressor genes are altered or inactivated due to mutation, they lose the ability to make protein that controls cell growth. Then two categories of tumor suppressor genes are there. First is caretaker gene or gatekeeper gene. In caretaker gene, maintain the integrity of the genome by repairing DNA damage, and in a gatekeeper genes, inhibit the proliferation of or promote the death of the cell with damaged DNA. This is the functional categories and tumor associations. The in gatekeepers, three genes are there. First is P53, RB1, or APC, and their functions are transcription factor, transcriptional regulator, or regulates beta catenin function. And in gatekeeper caretaker, there are three genes, BRCA1 and BRCA2. Their functions are similar. DNA, this is DNA repair. And third one is MSH2 and MLH1. Their function is DNA mismatch repair. Then their functions. Intracellular proteins that control gene expression of a specific stage of the cell cycle. If these genes are not expressed, the cell cycle does not continue. Effectively inhibit the cell division. Receptors or signal transductors for secreted hormones or developmental signals, then inhibit cell proliferation. Checkpoints controls proteins that trigger cell cycle arrest in response to DNA damage or chromosomal defects. The protein that induce apoptosis, if damage cannot be repaired, the cell initiates a programmed cell death to remove the threat it poses to the organism as whole. Cell adhesion, some proteins involved in cell adhesion prevent tumor cells from dispersing. Block block loss of contact inhibition and inhibit metasis. These proteins are known as metasis suppressors. Proteins involved in repairing mistakes in DNA. Caretaker genes encode proteins that function in repairing mutations in the genome, preventing cells from replicating with mutations. Certain genes can also act as tumor suppressor and oncogenes. The bed proto-oncogenes with tumor suppressor functions. These genes act as a double agents that both positively and negatively regulate transcription. And it is my conclusion. Suppression genes that are essential for the continuing of the cell cycle, and if the damage cannot be repaired, promote apoptosis, produce protein responsible for inhibition of metastasis and activation of tumor suppressor genes causes tumor. And this is my references. And thank you, sir. Uh, okay, okay, okay. Sir, APC or uh, regular uh, regulation of uh, functioning the uh, gene. P53.